Thank you to the organizers for putting this on. I know how much work comes behind a conference like this, and it's really an outstanding event. I know everyone's getting a lot of value out of it. So I am an inventor on some USF patents and intellectual property. I'm a scientific consultant for an exogenous ketone company, and I'm not a physician. So our lab at the University of South Florida is really interested in developing and testing non-toxic metabolic targeted therapies for a variety of disorders, but I'm most interested in looking at those for cancer. As you've heard this morning from some of the other speakers' presentations, many cancers seem to really thrive on having excess glucose and glucose signaling, but they seem to be vulnerable to energy stress. So we know that there's some evidence that shows that high glycemic index diets can increase the risk of developing cancer, that when you have hyperglycemia during treatment, you can have a poor clinical outcome. And in animal models, there's actually been some data to show a direct correlation between tumor growth and blood glucose, which has led other groups, as well as our own, to look at the ketogenic diet as a potential way to lessen basically this glycolytic phenotype of tumors. So a few years ago, we did some work in the VMN3 mouse model of metastatic cancers. It's a very aggressive model that metastasizes quite rapidly throughout the body. And we looked at two different ketogenic diets, both of which were able to slow tumor growth and prolong survival by about 50% in these animals. We did find that the survival of these animals was correlated to the ability to decrease tumor growth in them. But our lab is extremely interested in the role of ketones specifically. So we wanted to take a closer look. We knew that from the literature, there was data suggesting that ketones themselves may have direct anti-cancer effects. We showed it in some the VMM3 cells in culture when you add exogenous ketones onto varied levels of glucose, including even quite high levels of glucose, such as 25 millimolar, which is not atypical for cell culture preparations. You can get a reduction in the proliferation of these cells. This has been reported in papers back to 1979 across a variety of cancer cell lines as well. And when we look at ketones themselves, we know that there are some characteristics which may explain why they could be useful as an anti-cancer agent. So we know they stimulate oxidative metabolism. Many tumors really thrive on a glycolytic phenotype. We know they inhibit inflammation in part through inhibiting assembly of the NLRP3 inflammasome. They act as HDAC inhibitors, which we heard about yesterday morning. HDAC inhibitors is actually an entire class of anti-cancer drugs. So here we have an endogenously produced metabolite that may be eliciting similar effects. So they can alter the glycolytic pathways and even directly reduce glucose. Dr. D'Agostino mentioned this at the end of his talk yesterday. When we provide exogenous ketones, we see a reduction in blood glucose over time that really mirrors the elevation in ketones. So we looked next in this same model at a potent form of exogenous ketone, which is a ketone ester. And we saw that indeed, in this model at least, we did see actually a pretty similar reduction in tumor growth and prolongation of survival in these mice. These mice did exhibit some calorie restriction effects, so we included a calorie restriction control group and found that while they had a trend towards a benefit, it couldn't explain the entire benefit that we were seeing with the ketone ester. So in this model, at least, the ketone ester provided on top of a standard rodent chow was as effective as the ketogenic diet. So as I mentioned, we are interested in non-toxic therapies, and I think Dr. Champ made a really good point that these therapies often seem to work best when they're combined. So one thing that we wanted to evaluate was hyperbaric oxygen. So when tumors grow, they grow under very chaotic conditions, and the blood vessels that actually form within the tumor are very inadequate. So you see pockets of hypoxia that form throughout the tumor. This hypoxia actually stimulates a hypoxic response and dramatically promotes the aggressive phenotype of the tumor, promotes invasion and metastasis, reprograms metabolism, and confers chemoradiation resistance. So it's thought that restoring oxygen levels to the tumor could be a way to resensitize tumors to therapies that work via oxidative stress. And just a quick note on oxidative stress. Cancer cells exist in a state where they have elevated levels of free radicals, and that actually benefits them pretty greatly. So it allows them to increase mutagenesis, and they have an adaptive response in that way. These free radicals can actually also oxidize receptors for growth factor signaling pathways and promote growth factor signaling. But because they live at this elevated state, they're actually closer to a threshold where they can be damaged by oxidative stress. And so that's one reason many therapies tend to try to treat cancer via an oxidative stress-mediated mechanism. So hyperbaric oxygen is the delivery of 100% oxygen at elevated pressure, and this does increase the amount of oxygen in the tumor. So we then decided, let's combine these two non-toxic therapies, the ketogenic diet, as well as exogenous ketones in the form of the ketone ester with hyperbaric oxygen, and see what we can find. And we found a very dramatic slowing of tumor growth, slowing of metastatic spread, and the animals lived twice as long as control animals. So that was a couple of years ago. So what I really wanted to do was update you on what our lab's been doing since then. As I mentioned, we really think that ketosis has the potential to sensitize tumors to other therapies, and we want to look at other metabolic therapies as monotherapies, but also in combination. And so we have a number of other projects going on. I just want to give you a brief overview of those different projects. So first, we have some interest in trying to modulate tumor metabolism with glycemic agents. This was a project of Dr. Nathan Ward. He was a recent PhD graduate from our lab. He's now a postdoc at Moffitt Cancer Center. Brain cancer cells in particular really do have a robust glycolytic metabolism, and this makes them quite resistant to oxidative stress in general, but sensitive to energy manipulation. Two glycemic agents that we looked at in these cells were dichloroacetate, which inhibits pyruvate dehydrogenase kinase. PDK is the inhibitor of pyruvate dehydrogenase complex. So basically what happens is when you inhibit the inhibitor of PDH, you stimulate flow of pyruvate into the mitochondria. In many cancer cells, most of it, or a lot of it, is instead going towards lactate fermentation. So DCA will restore oxidative metabolism into the mitochondria of these cells. The other agent is metformin, which of course is classically known as an anti-diabetic drug, and we know that it inhibits hepatic gluconeogenesis, but it also has some molecular effects like activating AMP kinase, which would then inhibit PI3 kinase, AKT, mTOR signaling, which are all very active in most tumors to promote protein synthesis and tumor growth. And it also acts as a complex 1 inhibitor of the electron mitochondrial transport chain. And this can induce an energetic crisis and also induce oxidative stress. So first, in these brain cancer-derived cells, we found that DCA does in fact activate pyruvate dehydrogenase complex, and we can see that it causes the cells to reduce the amount of lactate that they're producing, so it's doing what we want it to do, which is good. And in a dose-dependent fashion, with increasing levels of DCA, we can reduce proliferation of these cells, as well as reduce viability. We also showed that these cells and this effect seems to be oxidative stress mediated because the effect can be rescued with N-acetylcysteine, which is a cysteine donor to help reduce glutathione. With metformin, we found that metformin was cytostatic to the VMM3 cells, meaning that it can slow or cease proliferation, but did not affect viability. When we combined these two agents, we found that even though metformin didn't affect cytotoxicity on its own, we can enhance the cytotoxicity of DCA by adding metformin. And this effect seems to be due to metformin's ability to inhibit complex 1 of the electron transport chain and generate oxidative stress. We know this because metformin in these cells did not activate AMP kinase. I'm not showing this data here. But we could actually mimic the same effects of metformin using rotenone. This is a complex 1 inhibitor, and we can't mimic the effects using ICAR, which is an AMP kinase activator. So again, suggesting that this effect that we're seeing in this cell line is probably mediated by complex 1 inhibition and not AMP kinase activation. And this effect can be partially rescued by an acetylcysteine antioxidant. So again, suggesting oxidative stress is probably a contributing mechanism. When we looked at each of these therapies individually, DCA and metformin at 200 mg per kg dosing was able to pretty dramatically slow tumor growth in these animals and each increased survival time. When we tried to combine the therapies, though, we didn't see any additive benefit of combining DCA with metformin. So it's possible that there are redundant mechanisms at play, or maybe in vivo we're seeing some kind of compensatory effect, which causes us to not see the same synergy that we found in vitro. Another study is looking at pharmacologic levels of ascorbic acid, or vitamin C, in the same model. So this study is the work of Janine de Blasi, a very talented undergraduate student who just graduated from USF, and we're losing her soon. She's doing a master's at Oxford, so I can't really blame her, but we will miss her greatly. Oral ascorbate can only provide micromolar elevations in the blood of ascorbate. But when you deliver it by IV, you can actually get quite high levels, or pharmacological levels, up to 20 millimolar. We all know of ascorbate's antioxidant effects at these low levels, but when you get to these very high pharmacological levels, you can actually elicit a very potent pro-oxidative effect in the tumor, and it doesn't seem to have the same effect in healthy tissues. And for the reason, this is under investigation in many different cancers. There's a few different potential mechanisms of action at play here. So for one, we know that ascorbate can undergo pH-dependent autooxidation. So in the tumor microenvironment, which is very acidic, ascorbate will auto-oxidize, which generates hydrogen peroxide. This can stimulate oxidative stress, which may be useful to damage the cancer cell. That oxidized ascorbate can also be taken up by the glucose transporters into the cancer cell. This has a very similar structure to glucose. Once it's inside, though, that oxidized ascorbate is often reduced by glutathione, and it results in an intracellular depletion of glutathione, which could cause these cells to now be quite sensitive to pro-oxidative stimuli. And ascorbate also is known to activate HIF hydroxylases. These are responsible for that hypoxic response that causes hypoxia to really dramatically increase the aggressive phenotype of cancers. So in vitro, we found that ascorbate, with increasing concentrations, you get decreasing viability and proliferation of these cells. So again, in a dose-dependent fashion. The effect on these cells seems to be oxidative stress mediated, which is consistent with the literature, again, because N-acetylcysteine can attenuate this decrease in viability. And we can mimic the effects that we're seeing with ascorbate by simply supplying hydrogen peroxide, so both suggesting this is probably occurring by oxidative stress. So we wanted to know, can we enhance ascorbate-induced cytotoxicity with hyperbaric oxygen? So we're all about trying to combine these non-toxic therapies to get a more robust response. And the answer is maybe in some cancer cells, but not in others. So we found that hyperbaric oxygen was able to enhance the effects in our VMM3 cell line, which is a brain cancer cell line, but not in U87, which is another brain cancer cell line. And we actually think we may know what's going on. It's possible that this difference in the response is due to a difference in transferrin receptor upregulation. So ascorbate will increase transferrin receptor in the VMM3 cells, but not the U87 cells. Transferrin receptor will cause an uptake of iron into the cell, which can stimulate Fenton redox biochemistry. And this Fenton reaction will cause the generation of hydroxyl radical and other very damaging ROS molecules. So having an upregulation in transferrin in response to this treatment could make those cells more susceptible to a large pro-oxidative stimulus compared to the U87 cells, which didn't see a compensatory increase in transferrin receptor. The next study that we're looking at is something called press-pulse metabolic therapy. This is an interesting idea. Basically, tumors they behave in many ways that are quite similar to ecological populations. And it's led researchers to ask, can we apply some of the ecological principles of population extinction to find new ways to target cancer? So press disturbances, these are chronic environmental stressors, such as maybe famine. Pulse disturbances would be acute events that cause high morbidity, like maybe a pathogenic insult. In ecological populations, when you have simultaneous press and pulse disturbances, that's when you see mass extinction events. So we think that ketosis may be a press disturbance for cancer. It's putting a lot of pressure on the energy pathways that really seem to promote robust growth and resistance in the tumor. And having a state of ketosis can really reduce many of those benefits that the Warburg effect bestows onto the tumor. We think this could actually be one reason why we saw this synergy between ketosis and hyperbaric oxygen. We can view ketosis as a press disturbance and hyperbaric oxygen potentially as a pulse disturbance. And so we want to look into this further because we think it could really inform better ways of targeting the tumor from all sides. And hopefully, if we're looking at non-toxic therapies, we could really improve the outcome without causing excess side effects. So some of the press disturbances we're interested in are the ketogenic diet, ketone ester, 2-deoxyglucose, targets glycolytic metabolism, metformin, and then some pulse disturbances such as hyperbaric oxygen, high-dose ascorbate, DCA, phenylacetate, this targets glutamine metabolism, as well as chemotherapy and radiation would be massive pulse disturbances. So this is very, very preliminary. The study is being led up by Sarah Moss, who is a really great undergraduate researcher in our lab. And literally the first round of this just finished up last week, so I was analyzing the data on the plane. But what we found so far is we've tried many of these things independently or in small combinations in this MMTV, mammary cancer model. And so far, the only thing that's sticking out is the ketogenic diet did increase survival time by a small amount. But we're excited to see when we combine these therapies, are we going to have a synergistic effect? Because again, we very typically see that these therapies work best in combination. And finally, the last project I just wanted to give you a glimpse into is looking at modeling and mitigating cancer cachexia. So this is the work of Andrew Kutnick, who is a senior PhD student in the lab, really fantastic researcher, and he's here at the conference. You've maybe seen him around, and you should go up and talk to him about his work. He had a poster last night as well. So cancer cachexia is a multifactorial syndrome defined by an ongoing loss of skeletal muscle mass, with or without fat mass, that cannot be fully reversed by conventional nutritional support and leads to progressive functional impairment. So this is a big problem in advanced cancer patients. You have large amounts of weight loss present in up to 80% of these patients. This weight loss really adversely affects the patient's ability to receive, tolerate, and respond to therapies. And it's estimated that about 20% of cancer patients will actually die from cachexia itself. And there is no standard of care. So the first question that Andrew wanted to ask was, does VMM3 model exhibit cachexia? So there's a lot of model systems out there, but none of them really reproduce the entire syndrome. Our model has a lot of benefits. It's syngeneic with the host background, which means we can use immunocompetent animals. It's metastatic from a primary tumor. We can track it with luciferase imaging, and it's very logistically feasible. So we set out to evaluate some markers of cachexia, including a metastatic phenotype, fat and muscle wasting, anorexia, which is present in end-stage cachexia, inflammation, and a few other biomarkers. I have to go fast, so I don't have a lot of time. So indeed, this model, of course, does exhibit a very severe metastatic phenotype. By end of life, these animals do have reduced food intake or anorexia, which is consistent with cachexia. Over time, you see that lean muscle mass decreases over time in these animals. So here's gastrocnemius and soleus weight and quadricep weight decreasing. And then by the end of life, you have a very dramatic wasting of the fat mass as well. We see increases in inflammation, many of the same markers that are known to be occurring in human cachexia patients, such as TNF-alpha, IL-6, splenomegaly, again, suggesting an inflammatory response, and some clinical biomarkers like blood urea nitrogen going up, albumin, and total protein decreasing. So we can say, yes, this model seems to be actually a very good model of cachexia. And so the next step, which we have not done yet, is to see if ketosis can work as a mitigation strategy in this model. So we know that ketosis is associated with anti-inflammatory and anti-catabolic effects. There's even some emerging evidence that it could be anabolic in certain conditions. It activates the HCA2 receptor, which will decrease NF-kappa B signaling, which is known to contribute to proteolysis in cachexia. And we're going to try that ketone ester that elevates both of the primary ketones, BHB and acetoacetate. So in summary, non-toxic metabolic targeting agents are really a promising class of therapeutics. Their effects are going to be cell, cancer, and model type dependent. We really need to be looking at them in combination with other metabolic therapies as well as standard care. And there is significant metabolic heterogeneity within tumors, and it really requires more nuanced conversations about efficacy of any individual treatment. We think that ketosis could provide the background for a variety of approaches that together could put extreme pressure on the tumor while reducing the side effects on the host. So with that, thank you.